Welcome to video 4 of the Chemistry for Biologists series. Today, we're learning all about proteins, what they are and why they matter. Let's get started. What are proteins? Proteins are macromolecules made up of repeating units, monomers, called amino acids. These monomers are linked via condensation reactions, releasing water to form peptide bonds. There are around 20 different naturally occurring amino acids that combine in various sequences to create a vast diversity of proteins. All amino acids have the same general structure: a central carbon atom, alpha carbon, an amino group, NH2, a carboxyl group, a hydrogen atom, a variable side chain known as the R group. The R group is the only part that differs among amino acids. Some amino acids have R groups containing sulfur or selenium. The R group does not take part in forming the peptide bond. However, it plays a crucial role in determining the chemical properties of each amino acid. Polar or nonpolar, charged or uncharged, hydrophilic or hydrophobic. These properties influence how amino acids interact with each other, especially in the folded structure of the protein. Interactions between R groups affect the tertiary structure, 3D shape, of the protein. Bonds in proteins. Peptide bonds, strong covalent bonds between amino acids, formed during protein synthesis, join the amino group of one amino acid to the carboxyl group of another. Hydrogen bonds, form between slightly positive hydrogen and slightly negative oxygen. Weak individually, but numerous, contributing greatly to protein folding, example alpha helices, beta sheets. Easily broken and reformed with pH or temperature changes. Disulfide bonds, strong covalent bonds between sulfur atoms of two cysteine amino acids, stabilize the tertiary structure of proteins. Less common than hydrogen bonds. Important for permanent shaping, example, hair perming. Ionic bond, occur between oppositely charged R groups, side chains, of amino acids. Stronger than hydrogen bonds, but less common. Can also be affected by pH. Proteins have 4 different levels of structure, each contributing to their final shape and function. The primary structure is simply the sequence of amino acids in a polypeptide chain, held together by strong peptide bonds. This sequence determines everything about how the protein will eventually fold. Next, we have the secondary structure. This includes patterns like the alpha helix, which is a spiral coil, and the beta pleated sheet, which looks like folded paper. These shapes are stabilized by hydrogen bonds between the amino and carboxyl groups. Some proteins may also form random coils without a regular shape. Protein will eventually fold. The tertiary structure is a more complex 3D folding of the polypeptide chain, including alpha helices and pleated sheets. This structure is stabilized by hydrogen bonds, disulfide bridges, and ionic bonds between the R groups of amino acids. Globular proteins like enzymes often have this structure. Finally, the quaternary structure is found only in proteins made of 2 or more polypeptide chains. It describes how these separate chains are arranged and interact in space. Hemoglobin is a great example of a protein with quaternary structure. Here, you can see the 2 main types of secondary structures in proteins, the spiral-shaped alpha helix and the folded beta pleated sheet. Denaturation of proteins. Denaturation is the process by which a protein loses its native 3-dimensional structure due to the disruption of the bonds and interactions that stabilize it, without breaking the primary structure, the peptide bonds between amino acids. Causes of denaturation. Heat. High temperatures increase molecular vibrations, disrupting hydrogen bonds and hydrophobic interactions. pH change. Extreme pH levels alter the charges on amino acid side chains, breaking ionic bonds. Chemicals, alcohols and urea disrupt hydrogen bonding. Detergents interfere with hydrophobic interactions. Heavy metals bind to side chains, altering protein structure. Mechanical agitation, vigorous mixing or shaking can disrupt interactions. Loss of secondary, tertiary, and quaternary structure. Protein becomes biologically inactive, often irreversible, though in some cases it can be reversible if the denaturing agent is removed. Fibrous proteins. Fibrous proteins are one of the 2 main types of proteins. Their structure is highly adapted to provide mechanical support, tensile strength, and structural integrity in biological tissues. Fibrous proteins are long, narrow, and strand-like. Unlike globular proteins, they are not folded into compact shapes. When we look at their polypeptide arrangement, the chains run in parallel and are often linked by cross-bridges, which gives them a rope-like strength and stability. As for the tertiary structure, fibrous proteins usually have little or no tertiary structure. They rely mostly on regular repeating patterns of secondary structure, like alpha helices or beta-pleated sheets. One of their important properties is solubility. These proteins are insoluble in water, which is essential for their structural role. They stay stable and do not dissolve in bodily fluids. Functionally, fibrous proteins are perfectly suited for structural roles. They provide support, strength, and protection to various tissues. And finally, some examples of fibrous proteins include collagen, which strengthens connective tissues, keratin, found in hair, nails, and skin, and silk fibroin, produced by spiders and silkworms. Function and biological roles. Found in connective tissue, tendons, ligaments, skin, hair, nails, bones, and feathers. Their toughness and insolubility make them ideal for structural support, common in extracellular matrices and cytoskeletal elements. Collagen structure makes up about 25 to 35% of total protein in the human body. Provides tensile strength to tissues that undergo stretching forces, example, skin, ligaments, bones. Found in cartilage, cornea, tendons, and the matrix of bones. Primary structure. Collagen has a repeating tripeptide sequence of glycine XY, where X is often proline, and Y is often hydroxyproline. Glycine, being the smallest amino acid, allows tight turns in the collagen chain. Secondary and tertiary structures. Each individual polypeptide chain forms a left-handed helix, different from the standard alpha helix. There is no significant tertiary folding within a single collagen chain. Quaternary structure. 3 of these polypeptide chains, called alpha chains, twist together to form a right-handed triple helix. This unique triple helical structure is stabilized by many hydrogen bonds and covalent cross-links, making it extremely strong. Fibril formation. These triple helix molecules then align in a staggered pattern, forming long collagen fibrils. Fibrils then bundle together to form collagen fibers, which give connective tissues their strength and stability. Osteogenesis imperfecta. This is a genetic disorder also known as brittle bone disease. It's caused by mutations in genes that code for collagen. As a result, the triple helix does not form properly, leading to weak and defective collagen structure, reduced tensile strength in bones, and bones that are brittle and break easily. Globular proteins. Globular proteins are compact, generally spherical proteins with complex tertiary and sometimes quaternary structures. They are formed by folding polypeptide chains in such a way that the hydrophobic R groups face inward and hydrophilic R groups face outward. This folding allows globular proteins to be soluble in water, or more precisely, to form colloids in aqueous environments. Proteins are made up of amino acids, and each amino acid has a side chain known as an R group. These R groups play a critical role in how the protein folds into its final three-dimensional shape. Hydrophobic R groups are nonpolar, they repel water. Because of this, during the folding process, these R groups tend to be found on the inside of the protein, tucked away from the surrounding water. This helps stabilize the protein's shape by minimizing exposure to the aqueous environment. In contrast, hydrophilic R groups are polar or charged, meaning they're attracted to water. These are usually positioned on the outer surface of the folded protein, where they can interact with water molecules. This arrangement increases the solubility of the protein in the cell's watery environment. Globular proteins are large and complex molecules, and because of this, they do not form true solutions in water like small solutes would. Instead, they form colloids, special mixtures where protein particles remain suspended throughout the liquid, but they do not settle out over time. This colloidal nature is crucial because it allows globular proteins to stay evenly dispersed in biological fluids like cytoplasm and blood plasma, enabling them to carry out their functions efficiently, whether that's transporting molecules, catalyzing reactions, or supporting cell structures. Globular proteins perform a variety of essential functions in the body. As enzymes, like amylase and DNA polymerase, they speed up biological reactions. In transport, hemoglobin is a key example. It carries oxygen in red blood cells. For immunity, antibodies or immunoglobulins bind to foreign antigens to protect the body. Some globular proteins act as hormones, like insulin, which regulates blood glucose levels. And within cells, cytoplasmic matrix proteins help maintain cell shape and hold molecules in place. Each of these roles highlights just how vital globular proteins are to life processes. Hemoglobin structure. Quaternary structure, made of 4 polypeptide chains, 2 alpha and 2 beta chains. Total amino acids, 574 amino acids in one hemoglobin molecule. Chains are held together by disulfide bonds. Each chain wraps around a heme group, a non-protein component containing an iron ion, Fe². The heme group binds oxygen reversibly, allowing hemoglobin to pick up oxygen in the lungs, release it in tissues where it is needed. The arrangement of polypeptides influences how easily oxygen binds, affinity, how readily it is released, dissociation. Conjugated proteins. Conjugated proteins are proteins that are covalently or tightly bonded to a non-protein group, called a prosthetic group. The prosthetic group is essential for the protein's proper function. The combination of the protein and the prosthetic group alters the protein's structure, function, and solubility. These proteins can be globular or fibrous in nature. Examples of conjugated proteins. Hemoglobin, a globular conjugated protein, contains iron as a prosthetic group, in the form of heme. Each of the 4 polypeptide chains in hemoglobin surrounds one heme group. The iron ion in each heme group can bind to an oxygen molecule. This enables hemoglobin to transport oxygen efficiently in the blood. 2. Lipoproteins, formed when proteins bind to lipids. Main function: transport lipids, especially cholesterol, in the bloodstream. The lipid portion makes them compatible with cholesterol, while the protein portion allows them to remain suspended in blood, aqueous environment. Lipoproteins help transport cholesterol in the blood. Low-density lipoproteins, or LDLs, have more lipid and less protein. They deliver cholesterol to tissues, but high levels can increase the risk of atherosclerosis. High-density lipoproteins, or HDLs, have more protein and less lipid. They remove excess cholesterol from the bloodstream and return it to the liver, playing a protective role. So, HDLs are often called good cholesterol, and LDLs are referred to as bad cholesterol. 3. Glycoproteins, proteins with carbohydrate prosthetic groups attached, found in mucus, synovial fluid, and on cell membranes. Functions: water holding capacity. The carbohydrate chains retain water, making the proteins slippery and viscous. Provide lubrication, e.g., in joints and digestive tract. Form protective barriers, example mucus lining in the stomach. Resistant to enzymatic breakdown, carbohydrate groups make them less susceptible to proteases. This protects tissues like the stomach lining from being digested by enzymes. And that brings us to the end of our topic on proteins. Thank you so much for watching. If you found this video helpful, don't forget to give it a like and show some love. And of course, hit that subscribe button and turn on the notifications so you won't miss any future videos. See you in the next one.